Thank you. And now we're moving on to our next speaker, who is Dr. Mike Rahe. Rahe? You just told me how to say it right, and I butchered it. I'm sorry, Mike. So Mike is an assistant professor of immunology at North Carolina State University, and his lab research focuses on the pig's immune response to viral respiratory infections. I know, Mike, you got your DVM from Iowa State, and then you came to Minnesota for a PhD in Dr. Mike Murtak's lab. So, and then you went to Iowa State again. So now you're back you're at North Carolina. So let's see a little bit of your journey in immunology of PRRS virus. So thank you. You prefer this one? That one? Okay.
Good morning, all. So we're going to move from pathology into immunology, which I know are people's two favorite subjects in veterinary school, right? So this is great. But today we're going to talk about the comparison of the immune response to circulating PRRS virus isolates, and we're going to specifically focus on L1C.5. The majority of the work that's been done has been on characterizing that response. Perfect. Thank you. So to start off any great immunology talk, we're going to talk about basketball. And Mike Krzyzewski is one of my coaching heroes, and he has this quote, which I think is incredibly true, and it says, "Effective teamwork begins and ends with communication." And I'm sure on the basketball court that's really important. I think it's really important for probably most sports, for work, for life, but it's also really important when it comes to the immune response. And so here's my starting five. And so you have macrophages, dendritic cells, neutrophils, T cells, B cells, NK cells are on the bench at this point, but you might see them later in the game. And you know, we think about these cells and you see them there, right, with Matt's presentation of the pathology, and it's neat to be able to see these things kind of in action. But when you look at those slides and when you see clinical signs, I think one of the questions we have is like, how is all this working together, right? And how are these cells able to communicate in order to achieve homeostasis, which is what they're shooting for, right? They want that pig to get back to health. And is it just like intuition? Do these sense, right? Like what's going on there? And so that's what we're going to talk about here for the first half of this talk. And so the immune response communicates by ligand receptor interactions. That's literally what they're doing, right? You have a protein which is bound by another protein and starts a signaling cascade, which generally results in the production of another protein. And so that's how they communicate, through these ligand receptor interactions. And the first example of that is here on the left side of the slide. And so in the bottom here, I'm going to use the pointer because I think maybe we'll see that better. So we have this dendritic cell, which is phagocytosed this virus, and it's able to sense that it's got a virus because of its pathogen recognition receptors, different type of PRRS, but pathogen recognition receptors. And there's a variety of these that these cells use, but they have specific ones for specific, what we call pathogen-associated molecular patterns, or PAMPs. And so through those general receptors, they're able to say, hey, I've got a single-stranded RNA sequence here. As a result of that, I'm going to start making type 1 interferon. Okay, and so that's really the way that the immune response gets kicked off against our pathogens, right? You have a dendritic cell, some sort of a sentinel cell, a macrophage, that's able to recognize that, like, hey, I've got something different here. I'm going to start making cytokines. And those cytokines get bound to receptors, and they draw in other cells. And by drawing in those other cells, now we're hopefully clearing the infection before the animal even knows it's sick. Right, and so all this goes on in the background within our tissues, within our pigs. And so really important how these things interact. And so that's kind of on the innate side, how these things work. Now this is really important as we get into the adaptive side. And so that's actually what we're seeing here in the bottom right. And so these dendritic cells, again, after they've recognized, hey, I've got a virus here, a lot of them are going to drain to a lymph node, and they're going to present the antigen that they've taken up to lymphocytes, so things like T cells, right? And the way that that interaction occurs is, again, it's ligand receptor based. And so here you've got this dendritic cell, it's got antigen, this T cell with its receptor recognizes the antigen. There's a series of checks and balances to this. And ultimately what happens is the dendritic cell gives a bunch of cytokines to the T cell and says, go forth and proliferate. And that's what it does. And so when Matt's talking about given large lymph nodes, but you can see those with really any infection, that's because that's what happens in infections, right? We get an adaptive immune response. You get your T cells, your B cells, they proliferate, make more copies of things that recognize antigen, and then hopefully those cells help clear the infection. And so really that's what's going on. But these cytokines are the key, okay? Without these proliferation signals, you get one cell that's trying to fight the infection. That's not going to work, right? We need to proliferate. We need to make more. And in PRRS, we know that our pig's immune response, they recognize PRRS and they produce cytokines. We've known this for a while. And I want to introduce basically 3 different buckets of cytokines here. The first one is the acute inflammatory cytokines. And so you might remember these from vet school or maybe in some research that you've done. So IL-1 beta, IL-6, TNF-alpha. These are kind of your early response cytokines, which are incredibly important in terms of response to infection. And we'll talk about them here in a second. The second bullet point there is type 1 interferon, and that's great because depending on which PRRS virus paper you're reading, either PRRS inhibits that response or you get a great type 1 interferon response. And so it's a gray area. And then type 2 interferon is interferon gamma, and that is probably as close as we've gotten at this point to be able to say we have some understanding of what helps clear PRRS infection. So there's been a number of studies which have shown that interferon gamma actually correlates with reduction in viremia or clearance of virus from the lung. And then the last one there is anti-inflammatory IL-10. We won't get into the depths of that today. There's been, again, another number of studies that have shown that with PRRS infection, you do get an IL-10 response. And so I want to highlight this figure. This is from a paper from 2010 from Joan Lunny. And here they're looking at the response to VR2332 infected pigs. And we're looking at IL-1 beta, so one of those acute inflammatory cytokines. And as you can see, day 7 to day 14, we see this uptick in IL-1 beta in the serum in these infected pigs. And by day 28, day 42, it's significantly reduced. And IL-1 beta is a big driver of fever. And if you think about when we tend to see fever in infected pigs, right, it's right at those time points. So this all checks. And the reason I took you through the walk through ligand receptor interactions and cytokines is because our cytokines drive a lot of the clinical signs that you all see with PRRS, okay? And so if we focus on those first 3, IL-1 beta, IL-6, or TNF-alpha, right, they're going to drive fever, lethargy, inappetence, vascular leakage, and if there's too much of it, systemic shock and death, okay? And so if you think about it, it's like, well, I guess I have a 3-year-old, so I get asked why about 40 times a day. And oftentimes when we're talking about this stuff, I ask myself, why, right? Like, why do we have fever, lethargy, inappetence, vascular leakage? Like, why can't the immune system do that without this type of clinical sign? But if you think about these things, right? So fever, we drive up the temperature. A lot of our viral proteins can't handle that, so they degrade. Lethargy in a herd situation, right? You don't want a sick animal being around everybody else and spreading it, so it makes sense that it would isolate itself. Inappetence, there's probably a number of different reasons. Either you're trying to starve out the replication of this infectious organism, or you're trying to prevent your pig from getting a GI disease on top of whatever else it's got. And vascular leakage, right? Where are most of our proteins that affect on these infectious agents? They're in the vessels. How do we get them out to where the infection's at? You have to open up those vessels and allow that fluid to get out and those proteins to get out. And so on a localized level, this is great, right? I mean, you cut your finger when you're working on your mower on the weekend, right? It gets red and inflamed. After a week, it's gone. On a systemic level, right, that's not good, because when you get systemic vascular leakage, now suddenly you go into DIC and you're dead. And so it's this delicate balance between these cytokines. And so I want to talk first by going through 2 different papers, which I think are fantastic. One came out in 2023. It was J.Q. Zhang's group, along with Marcelo Almeida at Iowa State. And with this paper, they looked at L1C.5, and they compared it to a number of different isolates that they'd seen in the Midwest. And I think the big one here is going to be in red, and that's 174, or lineage 1A virus, right? That was kind of the previous worst actor, and now we're comparing this L1C.5 that came along. And so they compared these infections in naive pigs, and if we look at their figures, you look at anorexia and lethargy scores, and L1C.5 blows the other isolates out of the water. You look at their temperature data, especially here from about 7 to 10 days of age, more severe fevers. You look at average daily gain, right? These pigs are losing 1/4 pound a day, where the other isolates are either holding steady or maybe losing just a touch less than that. And so really what this showed is what you all saw in the field, right? L1C.5 is very virulent. And so we know it's really bad compared to what we've seen in the past here in North America, but what about other PRRS virus isolates? And so this paper isn't a paper yet, actually. This is a preprint from the USDA. This is Jane Wiarta and Alexa Buckley's work. And they compared L1C.5 to, I apologize, Matt, it's high-path PRRS. And so this was an isolate from 2006, which got named high-path PRRS. And they wanted to see basically, that was kind of the gold standard in terms of the world, right? The most virulent isolate that we'd seen. How does that look? And so fortunately at the USDA, they have a BSL-3 facility, so they could do this. But you can see here, if we look at temperatures, your L1C.5 is right on top of that high-path virus. You look at body weights, the same. Maybe in the subjective scores, the lethargy scores and the respiratory scores, the red line, which is your high-path PRRS, is a little bit worse. But L1C.5 is up there as well. The other interesting thing about this study is they compared it to MN184, which many of you probably saw a few years ago or several years ago. And you can see there's really no comparison between L1C.5 and MN184. So this is a pretty bad actor. And so we walk through that. We walk through, right, the cytokine piece. We walk through the morbidity and mortality of L1C.5. And really where we're trying to get to is the linkage of, does the cytokine response drive a lot of the morbidity and maybe the mortality that you're seeing? And so in the paper out of the USDA, they did a really neat Leman assay on the serum because they wanted to look at the inflammatory cytokines over time and see if that also tracked, right? If the high-path PRRS was the same as the L1C.5 virus. And so they looked at type 1 interferon, they looked at TNF-alpha, IL-1 beta, and IL-6. We're going to skip over IL-4 that just got caught in my screenshot. And as you can see, right, L1C.5 and this high-path PRRS give you a really robust type 1 interferon response. They get this really bad TNF-alpha response, especially here around day 10, which is when they saw a lot of mortalities with these viruses. Also IL-1 beta and also IL-1/6. And so you actually see these cytokines peak right as a lot of these animals are about to die. And so it seems like there's a pretty strong correlation here between the cytokine response and morbidity and mortality from these viruses. And so what causes the difference in cytokine response, right? So we know these cytokines are kind of driving our morbidity and mortality that we're seeing, but why? And it's a fairly complex answer, and I don't have all of this worked out for you today, but I want to focus on a few things here to start. And so first, virus replication in numbers. That's what we're going to dive into because, again, we know our cytokines are driven by ligand receptor relationships, right? So if you have more virus, does that result in more cytokines? There's other possibilities, and so we talked about the pathogen recognition receptors at the beginning, right? Is it possible that these isolates have different targets for those? Are there different PAMPs? And so could that drive more of an inflammatory cytokine response? Is there some sort of interference with signaling pathways in the cells that these viruses can induce, and maybe that drives some of this response? And then the last one I threw in here just because I think it's kind of interesting, but these virokines, this is a DNA virus phenomenon, but in some of these DNA viruses, they've shown that they actually will make proteins that will act like cytokines that enhance their ability to survive. RNA viruses haven't been shown to do that, but I just thought it was interesting. What we're going to focus on is virus replication in numbers. And so if we go back to that USDA paper, right, we've shown that this L1C.5 and this high-path PRRS, they do a really good job of inducing this highly inflammatory response. What about numbers in terms of virions? And so if we look in the serum, again, our high-path PRRS is in red, our L1C.5 is in green. There's a little bit of separation there, but they're basically on top of each other. They cause a really severe viremia. If we look in the lung and bronchoalveolar lavage fluid, it's kind of up and down. It's a little bit more challenging to evaluate this, but they're basically on top of each other here as well. And so you get a really severe infection in the lung, also systemically. And going back to the Iowa State paper, here comparing, again, L1C 0.5 against the other isolates that they'd previously looked at. And so day 2 after infection up to about day 7, you can see a more severe viremia with L1C 0.5. They did that in vivo, and they looked in vitro as well. And basically, the results, if you want to summarize them, is L1C 0.5 grows really well in pigs. It also grows really, really well in macrophages. And Matt had brought up the Malini et al. Study, and I found that one as well. They looked at infectivity of L1C 0.5 in comparison to a lineage 1A, right, in that lineage 9 virus. And I thought this was absolutely fascinating. So here they did this dilutions. They took infectious virus and they diluted it down to 10 to the 0. And so there you're looking at like individual virions per mil. And they showed that at that infectious dose, they could infect 4 out of 6 pigs at 4, 7, 11 days of age. And so it looks like this thing is maybe even more infectious than what we've seen in the past. Now, where that perhaps enhanced infectivity comes from, not really sure. But again, the take home is really that this L1C 0.5 grows really well. And so putting this all together, L1C 0.5 grows better in pigs than previously, than the other tested North American isolates that we've seen. The high level of virus and resulting cytokine response, I think, is at least part of the reason that we see more severe clinical signs with this. And there's one element here that I didn't talk about because no one has really studied this yet. But as you increase the amount of infectious virus in those pigs, you're probably also increasing the number of macrophages that are infected. And you're also causing more apoptosis and necrosis. And so you're then releasing more of these damage-associated molecular patterns, which the immune system can't really differentiate from infection, right? And so you're enhancing the amount of signals that are out there, resulting in more release of cytokines as well. And so it's basically this perfect storm or this cytokine storm, which is why we're seeing these really bad clinical signs. Okay, so that's the innate response. But what about the adaptive response? Is there something that can be done there? So do we see differences between L1C 0.5 and other North American PRRSV isolates related to the adaptive immune response? I realize this is a bit of a busy slide, but this is the pig adaptive immune response summarized. And so I want to break this down. We're first going to look at the antibody response, and then we'll focus on the T cell response briefly. And so just a little bit on the PRRS virus antibody response, little summary. So from what we know, IgG seroconversion to PRRS typically occurs by 14 days post-infection. And this can be measured via ELISA or IFA. Neutralizing titers show up much later. Again, they're generally specific to whatever the animal was injected with. And so day 21 on depends on which paper you read, but that's probably a good average of when we first start to see a neutralizing titer. And these are measured with neutralizing antibody assays, or as it's shown in the next slide, FFNs. And so if we go back to JQ's paper, they basically saw no significant inhibition of the antibody response to L1C 0.5. And if you look down at their ELISA results, with blue being our L1C 0.5, right, we're right in the middle of the pack here. Same for the IFA response. And if you look at the neutralizing antibody response, you actually get a decent neutralizing response against L1C 0.5 by about 21, 28 days post-infection. Those are low titers, but it's occurring, right? It's kind of what we'd expect with PRRS. Okay, so that's what we know about the antibody response. What about the T cell response? And so I really want to focus here. We won't really talk about cytotoxic lymphocytes. I really want to focus on our T helper 1 interferon gamma producing cells, because again, that's what's been previously shown to correlate with reduction in viremia and clearance of infection. So it's likely that MLV-induced partial protection from challenge is coming from T cells. There's been a number of studies that have suggested or pointed to that. Which T cells, I think, is still the question, right? Is it CD4s? Is it CD8s? Is it both? It's probably both. But when we ran our L1C.5 challenge in my lab, one of the things we wanted to look at was interferon gamma expression in the lung, right, to see if that could correlate with reduction in viremia. And so we're wrapping up this study, but here in the lung, these blue areas, these are nuclei. The red is interferon gamma expression, and you can see we've got a high expressor there, and then maybe some other cells that are low expressors scattered throughout. Yellow is IL-10. And what we've been able to see through this is that we get a fairly robust interferon gamma response to L1C.5 infection. And the way that we're able to quantify that is we actually partner with Nikon and utilize AI, and they're actually able to parse this out and give us quantitative results on this. So be able to say, hey, you've got this many interferon gamma expressors or this many IL-10 expressors. And so it takes, again, a somewhat subjective assessment to an objective analysis. One other study I want to point to is this study which we are wrapping up and is in revision right now. And so in this study, pigs were vaccinated with MLV as well as MLV with Flex, so MLV with PCV2. And then they were challenged with lineage 1A 174. And this study took place, I think it was 2022. So this was kind of as L1C.5 is really getting on people's radars, and we're seeing it more. And so we did ELISpots for this T-cell analysis. And so here you take peripheral blood mononuclear cells isolated from pigs, and you incubate them with a particular virus. You basically run an ELISA thereafter, and you're able to see individual cells that recognize the virus that they were incubated with. Okay. And so that's what we're looking at here in these figures. So the green boxes are going to be individual pigs, their cells that were incubated with this 174 virus. And so as this goes up, that's saying, hey, there's more cells, more T cells from this pig that recognize that virus. The 144, well, it was 144 at that time. It wasn't L1C.5 yet. But we had this, again, this emerging strain, and we thought, well, let's throw it in. Let's see how the vaccinated pigs do to that. And so if we look at both our MLV and Flex, right, those are both pigs that saw MLV vaccine, you can see, first of all, that at 14 days post-vaccination, we've got a decent response against this 174 virus. Now, they had never been exposed to the 174 virus, right? They'd only been vaccinated. But even at this stage, you had T cells that were basically able to respond to a 174 virus. Similarly, they were able to recognize a 144 isolate. And so again, they'd never seen that isolate, but there were T cells, right, that were able to recognize an epitope on that vaccine and then also recognize the same epitope on these wild type isolates. And so I think that's really, really interesting. And if you forward, to the end of the study, this is 21 days after our pigs had been challenged with the 174 virus. I think this is also particularly interesting. So we get this nice boost from our vaccinated pigs, and they cleared virus better than the mock vaccinated pigs. But here, 21 days after challenge, you've got pigs that had not seen vaccine. They'd only seen 174 virus, but they're able to recognize both 174 as well as partially recognize 144. And so again, there's some level of conservation of T-cell epitopes between these isolates. So this is the last study that I'll bring up today, but it's bringing this all home. And so there's in 2024, JQ's group, they presented at AASV on comparing the efficacy of commercially available PRRS virus MLV vaccines to L1C.5 challenge. And so I just grabbed a table from their proceedings because I think this is important. And so if you look at these three vaccines in the red box, these vaccines significantly improved anorexia, lethargy, mortality, average daily gain, as well as viremia in those pigs. And so it seems like, again, it's not perfect, right? We didn't get complete protection, but there's at least some level of protection afforded by at least some of the MOV vaccines that are out there. Okay, so to summarize all that up, there doesn't appear to be large differences between L1C.5 and other PRRS virus isolates in terms of the generation or of functional adaptive immunity, so antibody response or the T cell response. And there does appear to be some level of cross-protection afforded by some of the MLV vaccines. And we think that this likely occurs due to the memory T cell response. Greatly appreciate the opportunity to talk to you all today, and happy to answer any questions you have.
Great talk to review the immunology. That's helpful. Thank you. The question I have is when we see these L1C5 or L1C2s and we've got naive pigs and they're getting laterally exposed and we get 70% death loss, is that the cytokine storm or what do you think's going on with that pig?
Yeah, it's hard, Paul. So when we ran our L1C study, we lost most of our pigs about day 10 after. I don't know if you all think it's about the same situation. We also had really bad neurologic disease in those pigs, and so I think that's the other component. It's hard to say which one, right? Because you've got massive pulmonary edema, they're in respiratory distress, and now they've also got neurologic disease. So it's I don't know if it really matters at that point outside of the fact that it's this virus that does it probably better than any we've seen. Yeah, I think you probably got a combination of things. Yeah, but the cytokine storm for sure doesn't help, right, when you're flooding the lungs with fluid and yeah.
Mike, a great talk. A quick question. From all those immunological predictors, if we think about the ones with available diagnostic tests, which ones you think it's still going to be the best predictor of protective immune response in the pig?
Yeah, I mean, I think the hard part is the prediction piece, right? Of like, what are they going to get infected with? You know, we're trying. You know, I'll back up. You know, Toby Kaiser was at NC State before me. He took the T cell response to PRRS and put it in the fast lane. And if you look at the data, right, and this isn't just him, this is other labs. They've shown that there are T cells that will react to different epitopes on different isolates. And so we really feel like that's where we need to be focusing. I realize that flies in the face of what I did my graduate work on, right, which was the antibody response. But I really feel like that's probably where the next steps are at. And that's going to be, there needs to be a lot of basic research to figure that out before we're able to take it forward and say, yes, this is predictive of protection. But that's at least the hope for our lab, yeah.
I just want to go back to that same example, but we're going to reverse it to the 20% that survive and say, what are those pigs doing different? Are they turning something off? Is there something different in the immune response?
Yeah, it's a great question, right? And so is it immune? Is it dose? Is it route? Like, there's so many questions there, Paul. I don't know. I'm not sure we've dug into those pigs yet and said, you know, yeah, what's different about you all? That probably takes a geneticist, which I am not, and you know, classifying MHCs and seeing if there's something related there too. But I think it's a great question. Yeah, what can we learn from the survivors from this? Yeah.
So, help me understand one thing. So, since this virus is replicating in the macrophages, who are the antigen processing cells to develop an adaptive immune response, so that means that some macrophages survive this replication. So to process some antigen and develop these cytokines. So in the in vitro studies you've seen, is there a difference in the survival of these macrophages to the replication?
Yeah, it's a great question. And I think, you know, so pulmonary alveolar macrophages, right, are the kind of the main target. Now it probably hits the pulmonary intravascular macrophages hard. There's dendritic cells that it probably doesn't infect. And those dendritic cells are really the keys, right? So they're going to be the big ones that take antigen and drain to lymph nodes and kick off the adaptive immune response. But I think, you know, honestly, as I was working on this talk, and I'll give away a research idea because maybe it's a bad one, but one of my thoughts was like, well, maybe we need to, you know, we have great sequencing technology now. You know, could you infect macrophages with L1C.5 and some of these other isolates and then sequence them and say, okay, what's different here, right? And is there any sort of a therapeutic route where we can hit this thing? It'd be in vitro, right? So maybe it'd be a little bit faster than run an in vivo study, but I think there is stuff that we can learn in vitro, especially with the technologies we have available now. So it's a great question.
Great talk, Mike. Thank you. Do you think immunity in some way could favor the emergence of higher virulent strains?
Oh boy. Yeah, so antigenic drift, right, is always a question. I think, you know, the big thing, and this is what I'm hoping to learn from Kim's talk, is is how different are these things, not just across GP5, but other parts of the virus? And is that really where this enhanced, virulence comes from? And so, while I'd love to be able to give you a more solid answer, I guess I'm looking forward to learning more about that too. It's a great question.
Hey, Mike. Great talk as always. I'm wondering if you have any thoughts on, from a diagnostic perspective, cell-mediated based assay that can be worked up to be offered and to understand better those survivors or those cell-mediated immune response from a diagnostic lab perspective.
Yeah, it's a great question, Fabio. It's one we've gotten before, and we're working on it. One of the beauties of antibody assays is serum is good for so long, right? And you can ship it, and it's still fine. And I mean, you still got to keep relative cold chain and all that. But one of the hard parts with doing cell-mediated work is you got to keep cells alive, and that's a challenge. We're getting better at that, and our shipping is also allowing it to where you can overnight samples. So we've been working on that in our lab of how do we make it so we get a viable sample that we can then evaluate in some of these assays. So ELISpots are great, but can we also take that to the next level and give you a better characterization of the cells that are responding? Now, the key piece there is we need to be able to do the basic research to be able to tell you that, hey, you need CD4s or CD8s because that correlates with protection. But ultimately, that's what we're shooting for is, hopefully not in the distant future, but in the near future, being able to provide that. Yeah.
I think, I'm sorry. Important thing is we need to know what is the protective antibody is. So far we know that the infected pig, viremia disappeared, and then we start to see neutralizing antibody. So, we've done very poorly on neutralizing antibodies.
Yeah, no, you're 100% right. And I think, just watching Danielle's presentation today, right, of MLV versus LVI, right? And so, with MLV, right, you don't get as bad of losses, but with LVI, I think the time to stability was shorter. And so the question there is, we know neutralizing antibodies take a while to develop, but maybe they're the key. So if T cells can keep disease at bay, are neutralizing antibodies the key at finally eliminating the virus? And so, that's something that I think we still need to investigate, but yeah, I think it might be important for the future.
Great talk, Mike. So your data, you talk about MLV potentially, increasing the ability of pigs to respond to viruses that they haven't even seen before. Danielle's, data showed that MLV introduction helped to reduce pig losses. From the immunologist perspective, pathologist perspective, virologist perspective, is that worth continually flooding the population with live virus when you have the ability to create new variants from recombination and other factors that go along with putting live virus back into the population? What are your thoughts on that?
It's a great question. I think, it's similar to what Danielle got earlier, and I think it's one of the tools that we have at our disposal now. Now, ultimately, we'd have a completely protective vaccine down the road that's hopefully non-infectious, right? That would be great. But with what we have right now, you all got to use it. So, yeah, but it's a great question.
Mike, thanks for the great presentation. So my question is, can you give a comment about killed vaccine? So what kind of protection will it confer? And what kind of practices would you suggest? For example, like MLV first and then followed by killed vaccine, or LVI first and followed by killed vaccine. From the perspective of the immunological responses. Any thoughts on that?
Yeah, no, it is another great question. It's one I've talked to veterinarians about before, and I think it's a big bucket, right? When you say killed vaccine, what does that mean? Because as far as I know, there's not a commercially available killed vaccine out there. Now, a lot of people are doing autogenous stuff. I don't, I'm not going to get controversial here, but, you know, I think there's oftentimes questions, or at least I have questions on efficacy. I'm not saying they don't work, but I'm just saying, you know, do they work? And I think testing those, I think, is really important. I think, you know, the idea of, hey, can we reduce some of the infectious virus on farms by using a kill product to boost? Like, I love it, but does it work, right? And I guess I haven't seen great data on that. And the hard part with that is if you were able to show it, does that translate to other people using different autogenous too? So it's a challenge, but I think it's another great question. So, Paul, do you have a comment?
Yeah, I was just going to comment on the killed. If you know, there's limited work that would suggest that you get more neutralizing antibody boost, and so I'm just wondering if that's a potential pathway that may be helping.
Maybe, yeah. Maybe it does allow you to get back to stable quicker. I don't know.
Like for the killed vaccine, can I comment? Like,
Sure.
does it have you evaluated or have a chance to read that the killed vaccine has more homologous protection because it's suitable for the field strain? It has a. Do you have any comments on that?
Yeah, no, I think to Paul's point, right? So you might be able to boost a neutralizing antibody response with it. You know, the hard part with the killed vaccines, just generally speaking, is they don't induce a great T cell response. And so if you're relying on that to get protection, you're probably not going to get great cross protection that way. But again, I'm not sure anybody's evaluated that to truly assess it. So
Have a follow-up. So that's why I'm asking, do you use MLV first to get it as a prime? So, or in the majority of the herd, you have done LVI first, or otherwise the herd is already like 80% infected, so it's already primed. So as a killed vaccine as a boosted solution. So what do you think on that?
yeah, I mean, I guess I'm going to go back. You can do that, but I would evaluate it and see what you get in terms of a boost in your neutralizing antibody response. It'd be an interesting study. So yeah.
Mike, is there anything that we can learn from, you know, all these viral infections happening in humans and being more relevant in the more recent years, right? Like, we don't talk about antivirals and using antivirals in pigs. Is that unheard of? Too expensive? I mean, what are your thoughts on that?
Yeah, I mean, there's so many questions on that of, you know, efficacy and then also cost, right? And yeah, I'll be honest. One of the things I've tried to stay away from, at least in my research, is running right to the, you know, bypassing the actual race and trying to get to the finish line. And I think, you know, we could do a lot better in terms of our targeted therapies if we knew more about how the virus interacts with macrophages, and then also how the immune response really, you know, does an effective job. And back to, I think, you know, Paul's point on studying the 20% that survive, right? Like, I would love for us to have a greater understanding on the basic immune response and pathology to PRRS before we keep trying to, again, be the hero that hits the home run. I'm okay with a walk that gets me on base. So, yeah.
That's great. Thank you very much. Once again, let's thank Mike.